Hello my dear student, welcome to Arunavasir bio class. So today we discuss a very important chapter, that is the genetics, and we discuss it for our class 10 ICSE board. This chapter we already completed in some topics, but today we discuss a very important topic. This is the day 3. So in our day 3 syllabus, the Mendel experiment on the inheritance, that is we discuss in our first topic, and next we discuss about Mendels Law of inheritance, and third topic is the mutation. That is the most important 3 topics is there in the ICSE syllabus, that is we discuss, and this topic also is very important in your CBSE board and other boards also, because genetics is a common subject, common topic, so then general all the boards have also the same topic. So all of the students like Sayan, Cinderella, Shatani, Korniket, Shorashish, Gaurav, Rastim, Anubhav, Shuham, Shudipto, Dishari, Shomojit, Indrila, Ankon, Shampridhi, Devarun, Shomoditto, Shutanu, Srijita, Debolina, Nirupama, Ishan, Krishnashish, Aroshi, Shoresh, Roshmi, Promit, Debargho, Shaptorshi, Shomojit and Kaushini, all of the students are special. Warm welcome to our special session for the genetics. So at first we discuss the very important topic, that is the Mendel experiment on the inheritance. You already know that Mendel are the father of genetics, and they work the experiment on a particular pea plant. Their scientific name is the Pisum sativum. But Pisum sativum is the scientific name. So when you are written in your copy, that time you must be separately underline this thing, and always the genus name is start with the capital letter, and the species name, their first word is the start was small letter. So Pisum sativum, that means the pea plant, is the most important plants for the research of the genetics by the Mendel. So but why? Why Mendel select the pea plant, and what is the different characteristic features is there for the Mendel success for the pea plant? Number one, you know the pea plant, they have many varieties were available in the alternative forms of a character, like height. This is a character. They have the 2 forms. Some plants are very short, that means dwarf, and some plants are very tall. So alternative some characters also be there, right? And some like seed shape, they have some round shape, then some wrinkle shape. So they have lots of different varieties is there for the pea plant, which is easy to study the genetics. Next one, variety were available pure form, that be the true. That means tall, in there the capital T is there, or the dwarf, that time it is the small t is there. So pure line, not the hybrid, are available in the pea plant very easily. And third important point, peas are self-pollinated. Self-pollination is very commonly done by the pea plants. Easily it is happening. But artificial pollination also be easily tested on the pea plant. That time the emasculation are happening. That time the anther are removed, and the artificial pollination are happening by the experiment of the genetics. So it is also be very important reason for the study of the pea plant for the genetical study. So that is the most important 3 reasons for the selection of the pea plant by the Mendel. And it is the most important question for your ICSE and other boards classes. But we are till that the pea plants are the different varieties. So what is the different varieties present in the pea plant? Pea plants have the 7 most common varieties. Like the seed shape, they have the 2 alternative forms. One is the round, another one is the wrinkle. Round is the dominant form, so it is the expressible, and it is the recessive form, that is the wrinkle. Seed color yellow and green, pod shape inflated and the constricted, pod color green and yellow, flower color purple and white, flower location axial and terminal, plant size tall and the short. There is a 7 distinguishable characteristic features, and it is very important for the genetical study. And you know Mendels are cross the pea plants in the two different way. First time they cross by only taking the single characters, only single characters. That time it is called the monohybrid. Mono means the single. And another type of experiment, they taken the two alternative forms, two alternative characters. That time it is called dihybrid cross. So monohybrid cross, that is the most important. So they have taken only one single character. They taken the character height. These have the two alternative form. One is the tall, another is the dwarf. Same way you write down this monohybrid cross. At first you write down the phenotype, that is the tall plant. That point you write down the genotype, capital T. Same way you write down the dwarf plants and write down the small t, and in that format you get the capital T, and in here in the small t. When the hybridization or reproduction or fertilization happening, that time the two gametes are fused together, showing it is called the P generation, and it is called the fast filial generation, which is you get by the cross of parent generation. So what is the phenotypic character? It is a tall. Why? Because the capital T is present. Capital T is the dominating alleles which is described the tall characters. Again, you cross these two type of plants, that means the capital T, small t into capital T and small t. So now you cross between the two hybrid plants and you get the F2 generation. But you write down it is as like checker box. It is the checker box. It is you write down the female part, and in here you write down the separately in the male part. Separate box, capital T small t, in here also capital T small t. Only cross this thing. Capital T and capital T write down in here. Capital T and small t write down in here. Capital T small t write down here. But always you write down the dominating allele in the first position. Next one, the small t in here. But what is the characteristic features? Capital T is present. So characteristic features are tall, and capital T small t also capital T present. Show the expressible character at the tall in here. Capital T small t show also be the capital, that T, that means the tall. But in here the small t and small t, that means the not the dominating allele are present. So small t are described the dwarf plants. Show the characters are coming, that is the dwarf. So what is the phenotypic ratio? How much number of tall plants you will get? 3, and 1 is the recessive dwarf. Show the phenotypic ratio, that is the 3:1, and genotypic ratio. Capital T you get only 1, capital T small t you get 2, and small t you get how much number? Only 1. Show the genotypic ratio is the 1:2:1. That is the genotypic ratio of the monohybrid cross. In this format, Mendel doing the dihybrid cross. That time only they taken the two characters. One is the seed color, that means the Y and Y. Another one is the seed cell, that means the round, capital R and capital R. And their alternative form, that is the wrinkle and green. Wrinkle and green, they are taken in that particular format and cross this thing. And this is the parent generation, and it is the F1 generation. F1 generation, capital Y, small y, capital R, small r. Capital Y present, small r, capital R also be present. So their character is the yellow round character. Y for yellow, R for round. In the F1 generation, they cross between the two F1 plants. Show capital Y, small y, capital R, small r into same alleles, capital Y, small y, capital R, small r. That time you get this checkered box. They have the 16 box, and after the find out, you concluded that the ratio of the F2 generation of the dihybrid cross, that is the 9:3:3:1. Round yellow, 9, and that is the green and round 3, wrinkle green 3, and wrinkle green only 1. So 9:3:3:1 is the ratio of the dihybrid cross. Very important. You just after the class is finished, you complete this form, right, and you write down in the below what is their character. You always mind, if the capital or dominant allele are present, their character are expressed. Bear in mind this sentence. After the experiment, Mendel get the law. Mendel give him the law. Mendel that not himself given the law. We get a conclusion after their experiment. Study the experiment of the Mendel. We get the law of dominance. What is the meaning by the law of dominance? Out of a pair of contrasting characters present together, only one is available to express itself, while the other remains suppressed. In the monohybrid condition or monohybrid case, we find out in the F1 generation, that is, we get capital T and small t, that is the genotype of the plant, and phenotype of it, that is the tall. In that particular case, out of a one pair of contrasting characters, that means capital T and small t is there, only one is able to express capital T, that means they are expressed, so we are get the tall character itself, while the other remains suppressed, small t, the dwarf character are suppressed. So this law of dominance we get from the monohybrid cross. Law of segregation, also called the law of purity of the gametes. In that condition, the two members of a pair of factors separate during the formation of gametes. Capital T and small t we get in the F1 generation, but in the F2 generation, all these two gametes are separated and the new character are formed. So we get the genotypic ratio 1:2:1 in the genotypic ratio in the monohybrid cross. So capital T and small t, these alleles are segregated. This is separated. So this is called the law of segregation. Next one, law of independent assortment. It is ultimately getting this law by the dihybrid cross. In here, when there are two pair of contrasting characters, like yellow round, that means the two characters, the distribution of the members of the one pair into the gamete is independently of the distribution of the other pair. Two alternative characters, that means the shape and the color of the seeds, are crossed together and we get the F1 generation. But when the F2 generation are coming, that time these two characters are not disturb anyone. They are independently assorted out or separated. In the dihybrid cross, this is called law of independent assortment. This is the Mendels law, and this is the most important for our study of the genetics. And last topic in our today's class, that is the mutation. Mutation is a sudden change in the genetical level or the genes. So genes have a particular phenotypic character, so their characteristic features will be changed due to the mutation. And it is also be hereditably transferred from parents to offspring. Mutation, why the mutation are happening? Mutation are happening in the spontaneous form, that is called the spontaneous form mutation. Another one is the induced mutation, like the UV rays, like the X-ray, and different chemical agents also be the induced mutation. That time it is called the induced mutation. Like you get one example, that is a sickle cell anemia. That time one gene is mutated, so hemoglobin, this protein is not formed in the good condition. So that time the sickle cell anemia are happening. Radioactive radiation, this is a mutagenic agent which can cause the mutation and show they change the genetical structure. So this is the mutation. This is the genetics chapter. After genetics chapter we study, you all of the students in the ICSE class 10, please you complete your exercise in your textbook. After that I give you some notes and some homework. Please you contact with me for the notes and the homework. Okay, so today class is now completed and we completed a special chapter, that is the genetics, and tomorrow after that the next class we discuss some important question about the genetics and important some questions and important some words, that is important some terms also we will discuss in the next class. If any doubts, please you also comments. I just solve your problem in the next class. So thank you very much all of the students. I wish you the best of luck.